Good morning, friends. Today we'll having a lecture of BSc third year on the topic of cell wall. We all know that the plant cells are surrounded by cell wall, plasma membrane, but the cell wall gives a mechanical support which strengthen, which gives the shape and maintain the structure of the cell. Cell wall is non-living structure which forms by a protoplast, or you may call it as without a cell wall, a cell is known as the protoplast. Now, what type of chemical constituent is there in the cell wall? Particularly, it is made up of cellulose, hemicellulose, protein, and pectin. In the case of fungi, cell wall is formed of chitin, and in bacteria, it is made up of protein, lipid, polysaccharide, or peptidoglycan complexes. Protection is one of the major function of the cell wall, which is rigid and gives shape to the cell. Now we see the chemical composition of the cell wall. Cell wall is composed of variety of polysaccharide, that is carbohydrates, proteins, lipids, mineral, etc. The staining reaction of various plant cell wall, which see this table. It is divided into three parts. Number one, substance, chemical unit, and staining reaction. So substance is cellulose, chemical unit is glucose. How the stain reaction takes, which is violet. So there are 6 types of substances: hemicellulose, pectin, lignin, mineral deposit, and cuticular substances. The polysaccharides of the cell wall, which shows cellulose, hemicellulose, pectin compounds, and lignins. Cellulose is a linear, unbranched polymer consisting of straight polysaccharide chain made up of glucose unit link. Then pectins. Pectins are water soluble, heterogeneous in nature, many negatively charged galactonic acid residues along with D-glucuronic acid residues. Mannan is a homopolysaccharide of mannose, and it is found in the cell wall of yeast, fungi, and bacteria. Agar is there. It is a polysaccharide found in the cell wall of seaweeds mostly, which contains D-N-L galactose. Lignin. We lot of heard about this lignin. Lignin is a biological plastic and non-fibrous material. It occurs in mature cells and is made up of insoluble hydrophobic. Hydrophobic nature means they don't love the water. Exactly opposite to the hydrophilic. Chitin is a polymer of glucosamine. Next, cutin is also a biological plastic and it is made up of fatty acids. And the last one, that is minerals deposit occur in cuticle in the form of magnesium, carbonates, silicates, and more on. Now we move. This was the chemical composition. Now we move to the structure of the cell wall, which is differentiated in following types, that is primary cell wall, secondary cell wall and tertiary cell wall. The first cell wall is known as the primary cell wall, which is the outermost layer of the cell, and immature meristematic and parenchymatous cells. It forms only in the cell wall. Primary cell is comparatively thin and permeable. Cells of the leaf and the stem also possesses the cutin and cutin-like waxes, which makes the primary cell wall impermeable. The primary cell wall of the yeast and the fungi, we know that they are made up of chitin. Whereas secondary cell wall is followed by the primary. Secondary wall is thick, then the primary cell wall permeable and lies just near the plasma membrane. Latter occurs, it is composed of three concentric layers, that is S1, S2 and S3, which occurs one after the another. The secondary cell wall is composed compactly arranged of microfibrils of the cellulose, in between which sometimes occurs lignin as intrafibril material. And the last, that is tertiary cell wall. Certain plant cells, there occurs another cell wall beneath to the secondary wall, which is also called as tertiary cell wall. The tertiary cell wall differs from the primary and secondary in its morphology, chemistry, as well as staining material. Beside this, the cellulose, the tertiary wall consists of another chemical substance, that is xylene. Now, middle lamella. What is middle lamella? It is generally the plant tissues generally remain cemented together by an intercellular matrix, that is known as middle lamella. The middle lamella is composed of pectin, lignin and some proteins itself. Now we see the electron microscopic ultrastructure of the cell wall. Same architecture principle which applied well in the construction of animal bones and such common building material, as like we see the plastic plus glass fiber, concrete plus metal framework and so on. In primary cell wall, the fibers and matrix molecules are cross-linked by a combination of covalent and non-covalent bonds to form a highly complex structure whose composition is generally cell specific. You can see in the figure 5.28 how this ultrastructure is made up cell wall. What is plasmodesmata? In living cell, mostly in higher plants, plasmodesmata connects to the neighbor cell with the cytoplasmic channels, that is known as the plasmodesmata, ribbon-like ligament. This plasmodesmata helps to pass the intervening cell walls. Plasma membrane of one cell is continuous with that of its neighbor at each plasmodesmata. A plasmodesmata is a roughly cylindrical membrane lined channel with a diameter of 20 to 40 nm. Now, the lignification of the plasmodesmata. The structure of cell wall is stabilized by the deposition of the lignin in the cell wall matrix. Such process of lignification was required in connection with the transition from aquatic to the terrestrial plant life during the organic evolution of the microfibrils of cellulose embedded of the matrix containing large amount of lignin. And the last we see the functions of the cell wall. What is the function of the cell wall? We know that the cell wall is function as a strengthen, maintain the shape, or we may call it as like a skeleton of animals. The cell wall functions in the plants. It is one of the framework or skeleton of the plant. Despite it is permeable for water and solute, this is because the matrix is riddled with minute water fluid channels with free diffusion. Soluble substances such as gases, salts, sugar, hormones, and many more. The molecules of the matrix are strongly hydrophilic, that is, water loving, which result that in normal circumstances the cell wall is saturated with water like a sponge. So friends, these are the permeable functions of the cell wall which allows the water and solutes to pass through the plasmodesmata. Therefore, plant must subsist on molecules of lower molecular weight and any intracellular signaling of the known plant that such as growth regulating substances, auxins, cytokinins, gibberellins have molecular weight of less than 500 daltons, and some of the macromolecules they have the molecular weight of more than 20,000 daltons. Now, how the origin of this cell wall has been taken? There are few following steps. Number one, formation of matrix. Matrix components are transported via vesicles derived from the Golgi apparatus and secreted by the exocytosis in the plasma membrane. Perhaps following facts are there. Each of the polysaccharides of cell wall matrix is made up of two or more sugar. At least 12 types of monosaccharides are used in their polymerization. Most of the polysaccharides are branched. Many covalent modifications are introduced in the polysaccharides. Phragmoplast. What are phragmoplast? Phragma means H and plast means to form. Form in the plane of former spindle equator. The phragmoplast comprises a double ring of short microtubules on either side or at the terminating point. The division plane and a set of microfilaments are co-aligned with the microtubules. Golgi derived vesicles containing cell wall precursor, especially pectin, are guided inward along the orientated microtubules until they reach the phragmoplasts, where they fuse with one another from the cell plate. Now see, there are cell plate at this stage comprising some following structure, that is central middle lamella, primary cell wall, and third one that is plasma membrane. So friends, while seeing this cell plate, we see that there are three plates. Here you see the diagram of the model explanation of the model orientation of newly deposit of cellulose microfibrils according to the orientation. The second one is synthesis and orientation of cellulose microfibrils. Here you see the fibril deposition in this figure. Most plant cellulose synthesized on the external surface by plasma membrane which bounds enzyme complex called cellulose synthetase. Now, while this question may be asked in the examinations, we must be able to learn all these things to answer. So friends, synthesis, which uses a sugar nucleotide precursor supplied from the cytosols, probably precursor supplied from the cytosols, UDP glucose. As they are being synthesized, the nascent cellulose chains spontaneously assemble into the microfibrils. So this was regarding the cell wall, cell function, cell structure, and regarding the origin of the cell structure. The diagrams or the figures or the matter which is shown in this video is taken from the book known as the biology, cell biology, genetics, molecular biology by P.S. Verma and V.K. Agarwal. Thank you.